All right. So thank you so much, Marcy, for the introduction. Like you said, I'm Hojae, and today I'll be presenting my ongoing work on predicting neural morphology from single-cell gene expression using deep neural models. Matt and Crystal, I know you're in lab and have heard my lab meeting recently, but this is actually different content, so good on you. Thanks for coming. So I'm going to put everybody on a slightly uncomfortable position. I'm going to put people on the spot here before I jump in. So, sorry to interrupt your eating. Can I ask how many people may have attended Josh's DCMB talk? Maybe a fair number. Yeah. Can I ask, were masters, maybe? Masters, few masters, pre-candidate? Okay, one pre-candidate. Everybody else is candidate or above. Okay, sounds good. Faculty, everybody. Yeah, so just like a little disclaimer, if you have attended Josh's talk, there's going to be a slight overlap, but not too much. I tried to differentiate it as much as I could. This particularly, I'll try to focus on the tools and technology portion so that you can use the techniques here and have better understanding of the inner workings of the techniques, and also help you think about how you might want to apply to your own project. And I'm always welcome to talk about like different ideas and different ways to apply this. So feel free to come talk to me afterwards with your own ideas. And yeah. So without further ado, I'm gonna jump straight right into it. So the goal of this project is to predict the shape and structure of neurons based on their gene expression. So why might we want to do this? Well, first, neural morphology is a key component of neural circuitries that is responsible for information processing in the brain. Predicting from gene expression is particularly interesting because now we're kind of explicitly creating a model to study the relationship between genes and morphology. And this can be especially interesting if you're studying disease models like Alzheimer's or Parkinson's. We can now start to kind of investigate in silico the effect of gene expression on morphology. And historically, morphology has always been a key component in determining neuron cell type. And one of the goals of the Brain Initiative is to try to create a comprehensive compendium of all the cell types. And it's not going to be just achieved by transcriptomics alone. It's going to be some combination of transcriptomics, morphology, electrophysiology, etc. And I mentioned this in lab meeting, but I like to give like a little historical fun fact on my presentation. So this picture on the right is the first hand reconstruction, some drawings of neurons by Santiago Ramon y Cajal. I hope I'm pronouncing it somewhat correctly. So he is definitely one of the first persons to have done hand reconstructions of neurons. Now it's mostly done kind of digitally. So you can kind of see the extreme kind of skill that went in here. And actually, this is a perfect week to present this because, as you may know, there's the Nobel Prizes are being given out or awarded this week. And Cajal, along with Camilla Golgi, won the Nobel Prize for Physiology and Medicine back in 1906 for their work in elucidating the nervous system. So yeah, a very kind of broad overview of how we're going to achieve this is. So we are going to use some deep generative models to predict condition on single cell gene expression to predict neuromorphology. So there are several types of deep generative models, including GANs, VAEs, and diffusion. In this talk, I'm going to mostly focus on GANs and VAEs. So this is where I'm going to diverge from lab meeting a little bit. If you're curious about all these or any of these three, Josh and Gia's course, Bioinformatics 593, will soon delve into this topic, and I'll mention that a little bit later in my talk. And this is a nice time to talk about deep generative models just because there's a lot of public awareness and interest in it. So you may have heard of examples like Midjourney or DALL-E or ChatGPT. I made a joke, if you have not used ChatGPT or have not heard of it, like you should probably go there now instead of listening to this talk. I will not be offended. So feel free to tune out if that is the case. But these two particular deep generative models are really good at taking like the text information, like a photograph of an astronaut riding a horse, and somehow using that information to create an image that incorporates the semantic meaning of the words into image. So in my project, instead of thinking text to image, you can kind of think of the problem as how can we utilize information in the gene and create morphology based on that. So that's kind of a simple analogy to think about what we're trying to do here. Here's a brief list of what we're going to be talking about. I'll just give you straight up the overview of the model that we developed for gene to morphology prediction called MorphNet. I'll talk a little bit about the patch-seq data set, which we use to train the model. I'll talk on both the single-cell RNA-seq portion and also the morphology portion. I'll spend quite a bit of time on, maybe not quite, but at least a little bit of time on GANs and also style GANs, which is what MorphNet is primarily based on, some results and summary. And also feel free to stop me at any point as well. So here is the overview of MorphNet. So this model combines actually two generative models, one VAE and one GAN, and I'll go into each of them more deeply to take gene expression information and predict morphology. So the VAE, which is on the red box, is so the purpose is a little bit different. The VAE is used to obtain meaningful latent vector representation, and I'll explain what that is in a few slides. The generative adversarial network, which is in the green box here, is used to transform or to generate the image. Usually there's a starting image of noise, and then it will take that noise and also the gene expression to generate morphological images. Okay, so much more detail of both of these components will come soon. As far as the data set, I use a really unique kind of a data set called patch-seq, which is a trimodal data set that combines patch cloud recordings, which I don't actually use in this project, but that's where the patch and patch-seq comes from. Also the single-cell RNA sequencing and neuron tracing / reconstruction, all from the same cell. So this gives us the paired gene expression and morphology data. So compared to, I guess, other data set, there is a ground truth morphology here from the same cell. There's a gene expression and the ground truth corresponding morphology. This is a very, very brief schematic of what patch-seq is collecting. So either starting from a slice of the brain or a neuron culture, you can do some staining to get like a morphological reconstruction. You can also poke in some probes into the cells. It's very hard. I haven't done it myself, but I heard it's very hard to get like recordings like action potentials, not exactly, but something similar to that. And afterwards, you can suck out the nucleus from each of the cell to do the single cell RNA sequencing. So as you can imagine, this is quite hard to do. And so compared to other kind of single cell methods, this is quite low throughput. So it's I am using like two of the largest publicly available patch-seq data sets to combine together have like just over 1,000 cells. And I'm just mentioning this because it's one of the considerations when you're developing because I guess any kind of deep learning models, just in general, they require a lot of training data. So we do need to think a little bit about how we're going to deal with this limited training data. And if anybody is interested in this kind of morphology data set, Neuromorpho is an excellent repository that started in 2006, and I think it has just about quarter million morphologies, but not all the morphologies will have single cell components in. So this is more morphology focused data repository site. So I am going to also assume that many people here are probably familiar with single cell RNA-seq data, or what it looks like at least. I know that's not usually the case in other audiences, but in general, like we're starting out with this, cell by gene matrix or gene by cell, depending on who you are. In my world, cell by gene, where the individual elements are non-negative integers, so these are count data. I'm going to say that these are, quote unquote, high dimensional. especially if you're collecting data from the whole transcriptome, they can be, maybe if you open one of these files, it can have like maybe up till 30,000 genes or so. What we did in this project is that we fed this through a variational autoencoder that I'll go into detail to get a cell by gene latent representation. So compared to the original data, this is much more lower dimensional. In fact, we set it to 10. So instead of 30,000, we're kind of compressing all the information into a 10-dimensional vector using a variational autoencoder called SDVI. So the VA portion, we're building off of this work called single cell variational inference. The paper has been published already in Nature Methods. I think the author is now also professor in biostat, maybe, or in stat, I forget exactly. But he's also here if you want to actually talk to him, is what I know. So SDVI, which stands for single cell variational inference. Let's see how to explain this. So VAEs, it's a little bit confusing because VAE itself actually is made up of two different neural networks, one called encoder and one called decoder. So the encoder portion, what this does is it takes a high dimensional and learns a low dimensional vector. And then the decoder is taking the low dimensional vector and trying to reconstruct the original data. So I've taken this really big and kind of information dense figure. I tried to kind of segment it out to the relevant parts. So in their encoder, basically they have, two really important neural networks that will learn the mean and the standard deviation of a multivariate Gaussian. I've written that out here, kind of. And then what the decoder does is that it actually it assumes that the gene expression data is coming from this so-called zero inflated negative binomial or ZINB distribution. So it uses the information from the multivariate from the compressed data. So you can see like this data is actually used in this parameter for like expected frequency and expected dropout. That's kind of, so this is their model of where the gene expression is created from to actually reconstruct original data. And as you can imagine, you can train this model by comparing the original with the reconstructed. Obviously, the goal is to have them match, and through this process of trying to match that, it's going to try to learn a latent representation that's compact, so that the decoder can reconstruct properly. So that's kind of a nutshell of what's happening here. In the end, once the VAE is trained, we only use the encoder part to get the latent vector representation. Unless you are interested in some other, depending on the application, you can use your decoder or not. In my case, I just want to train the VAE and use the encoder to get a lower dimensional representation so that I can feed that into the GAN. The neuromorphology data set, probably not many are familiar or less familiar. So these are actually, the data is not actually given in images themselves. They're already digitally reconstructed by the data producing lab. And they actually come in this tabular format called SWC files, which contain 7, is it 7? 1, 2, 3, 4, 5, 6, yeah, 7 columns, ID type XYZ radius parent. So each row is representing like a point in space or node. The type is indicating whether that point is part of the soma or the axon or dendrite. So it's only a few values in there. And you can use your favorite, pandas or R tools to, read this data frame to create, and plot them to get like a 3D view, or in my case, in this project, I focus actually on the 2D view, something like this. So if you were to look at it from the up, down, you'll get this kind of like flattened image. So the goal of MorphNet was to try to predict the 2D images of neuromorphologies. And I just want to also say that this is like one way to think about neuromorphology. I'm thinking of it like the neuron structure as like a set of 2D images that you can possibly combine together to make a 3D structure. But there are many other ways to approach this that can change the underlying technique a lot. So you can think about neurons as like a bag of branches that you put together, or you can think about them as graphs, a particular like a tree graph, or yeah, and depending on kind of how you represent your neuron, kind of as a mathematical object, you kind of change the underlying method. Anyway, but for the purposes of MorphNet, we obviously chose the image representation because GANs have been so successful in generating high quality and high fidelity realistic images. Okay, any questions so far? And I'll give it, yes.
I'll transform the image to the image.
Yeah, so the question was, how do we transform to a 2D image? I literally plotted this on matplotlib and then saved it as an image without the axis. So without the bounding, like if you plot in matplotlib, you get the x-axis, y-axis. So you can just turn those off and you want. And so all the kind of coordinate positions are kind of lost in that sense. And we're just thinking of neurons at this 2D as just like a pure pixel representation. So like if you were thinking about this as like a square image, there will be a lot of white pixels, like 255, 225, 255, and there will be some pixels that represent red, black for soma, etc. Just so the model doesn't have any idea about like the points or the connectivity information. All right, I'm gonna move on for now. So background on generative models. Broadly speaking, kind of regardless of the generative model that you're talking about, whether it's GANs or VAEs or diffusions or normalizing flows, their broad goal is to learn the underlying data distribution of the real samples. So they're generally categorized, or one way to categorize them is whether they learn an explicit density model or an implicit density model. An example of explicit density model is like VAEs or probabilistic PCA. But who is taking Josh's class right now, if any? Okay.
Right now.
Yeah. I think he has maybe just gone over or like just on a probabilistic PCA lecture recently. And so the goals of these models is to learn this blue probability distribution. And you can imagine if you have this distribution, you can sample from the distribution to create new samples. One dominant example of an implicit density one is GAN. So GANs doesn't actually learn a probability distribution model, but it will learn to generate or learn samples that kind of that if you were to draw the probability distribution for the data points and also the model samples, that if you were to draw the curves, then they will actually match, but it will actually not learn the curve itself. Okay. So getting into GANs a little bit more, like the VAE, this also consists of two models inside, one called the generator, one called the discriminator. They all have different names, very confusing. And so a little bit historical perspective as well. So this was published back in 2014, kind of the early days of like the new kind of spring, hot summer of AI, I don't know, out of the AI winter. So if you remember, or I think we have just recently gained like a new interest in AI after AlexNet did very, very well in the ImageNet classification back in 2012. And it's not that, and the authors here were interested in how we can generate good images, not just classify them. But this generation process has always been quite hard. So the inspiration was, how can we kind of utilize this success and kind of powerful discriminator models like classifiers, so that how can we use this kind of as like a teacher to teach good teach good generator networks. So that's partially one of the inspiration. If you look at the original paper, I think it actually said like, how can we utilize the success of AlexNet to create good generator networks. Again, in a nutshell, these two networks are gonna play this minimax game where they're competing with each other. The generator is going to try to generate realistic looking data. The discriminator is going to try to it's going to do a binary classification, whether for a given image, whether this image is real or fake. So it's not a complicated classification. It's not like classifying an image as like dog, cat, and 1,000 other classes. It's just, real or fake. And actually the information from the discriminator, so whether it's real or fake, is actually gonna back propagate to motivate the generator to do better. And actually this competition is actually going to make both of them better at their jobs. The generator is going to get better at generating images. The discriminator is going to get better at telling the difference. Pictorially, what's happening is, I'm going to try to explain this as clearly as I can. Let's see. So these black dots represent the distribution of the real data. So that's the real kind of data that we want to mimic at the end. The green is the distribution of the fake, and the blue is just the decision boundary of the generator. This is for a simple 1D example. So, first thing that's going to happen, the generator is going to sample from this Z space. So let's say this is some noise. It seems like it's sampling kind of uniformly. Generator, like any other deep learning models, is just a function, like it's just going to transform one input to another, right? So it's going to transform the Z into this X space, let's say images or morphology or whatever. So you can see that's kind of concentrating the outputs here, which makes this, green distribution. So once a generator creates that, we can now take the discriminator to train whether it's whether a given image is real or fake. So in the beginning, it doesn't do very well. So it's all these squiggly lines, but in the once you train the discriminator, you'll see that like points coming from this end on the left is most likely real. Points coming from this end is most likely fake. And you can see there's a little slope here. So this gradient in multidimensional term is going to, you can almost think of it as like an invisible force trying to pull the green curve over to the left. I don't know if you can I can feel it from the picture, but think about, like, there's information here, there's a gradient here, and it's going to push the green towards the black dotted lines. So you can see once that updates, the green has come a little bit closer, and we're going to repeat this process until eventually the green very closely matches with the black, which is the original goal that we wanted. We wanted the generator, aka the green line, to match as closely with the black, the real distribution. Okay, so that's pictorially. This is more mathematically trained using, so GANs are trained using so-called adversarial loss. This one, I feel like it looks kind of scary in the beginning, but actually it's very, 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 very simple. So this left term here is the log probability that the discriminator correctly predicts the real data as real. If you drill it down a little further, you can see X is being sampled from the real distribution, PR of X, that's a real distribution. So, and that's going to go into a discriminator. The discriminator is going to say, it's going to give some probability of whether it's real or not. So it's going to give some value between 0 to 1. This value is maximized, so log of 1 is 0, right? And the log of 0 is minus infinity. So if the discriminator wants to maximize this value, it will want to try to classify this as real, right? On the flip side, this is the log probability that the discriminator correctly predicts generated data as generated. Again, the same process. If we sample some noise from PZ, so let's say PZ is some uniform or Gaussian, doesn't matter. The generator is gonna take that noise and generate fake data. That's G of Z. The discriminator is gonna see that, and again, it's gonna output some value between 0 to 1. Again, if the discriminator wants to maximize it, then you want this value to be close to 0, right? Because log of 1 is 0. Again, if this value is close to 1, then it's going to be log close to 0, which is very negative infinity. And the E here is, we're going to average this. That's a simple way to think about it. So you can already there's competing interests here between the generator and discriminator. That's the main takeaway. So discriminator's goal is to maximize the prediction accuracy. The generator's goal is to try to fool the discriminator because it has the opposite goal here. It wants to make this as negative as possible. You can actually solve this optimization problem, not shown here, 10 Bioinformatics 593. But after the math shenanigan, the optimal value is when the generated distribution PG of X matches the real distribution PR of X. Once this holds, obviously the optimal value for a discriminator is that's perfectly confused. It will just be unsure whether it's real or fake. And the loss, the value of the loss, which is actually not important, but if you're interested, the value of the loss will come out to be -2 log 2. Anyway, well, skipping a lot of details there about derivation, but we're gonna move on to bigger and better things. Okay, so the original, again, like I mentioned, I actually took the screenshot now, like it's been 3 years ago, so maybe like 2018-19. So it's going only from 2014 to 2018. A lot of things have happened since then as well. But this is just to give like a little context. So the original, again, was only 32 by 32. And since then, we have gotten higher resolution and higher quality. This is obviously not to scale. 1024 by 1024 is much larger than 32 by 32. And you probably, I always like give an example of like this website called like This Person Does Not Exist, which if you go there and refresh, every time you refresh, it'll give you like a picture of realistic looking images, but not real. So feel free to play around with that if you're interested. It's really cool. Again, in class, I do go over like all the intermediate ones before I get to StyleGAN, but for the interest of this talk, I'm gonna just jump right into StyleGAN. So the major kind of innovation for this paper was this humongous generator design. So it's not just your, I guess, convolutional layers stacked on top of each other. So they actually separated out the generator to this 2 networks underneath one called the mapping network, which is all this FC fully connected layers stacked here, and then the synthesis network, which is responsible for taking the noise image and generating the image. So since the synthesis is kind of like your traditional generator that we have talked about before. Yeah, and the mapping network, I guess, is more the novel part. And also like another innovation was that instead of just taking your initial like latent or gene expression in our case and going through a series of convolutional layers, StyleGAN actually tried to introduce the latents at every single layer of the synthesis network. So you're kind of inputting information across every single layer of the generator. Yeah, so that's kind of an expansion, architectural kind of expansion of what happened. I mentioned about training GANs with limited data. I think in general, you probably want on the order of 10 to the 5 or 10 to the 6, like up to 1 million images to train well, at least for natural images like faces. I guess it's a little bit unknown for non-natural images, but that's simply what's cited on literature. I would see is what I would see. But so the same authors who developed StyleGAN 1 tackle this problem of how we can generate good images, maybe not the best quality, but good images with limited data. And one of the things that we do when we have a classifier that is overfitted is to use data augmentation. I mean, there's many other methods to prevent overfitting, but one thing is data augmentation. A little bit tricky part with generative models is that you don't want to have the generator creating augmented images. Like for example, I have some like pictures of a painting that is, let's say you do some kind of color augmentation and you generate some blue faces, but you don't want the generator to make blue faces unless that's your application for some reason. We still want to generate like normal things. So brilliant idea. We're just gonna augment the discriminator only and not touch a generator. It's a little bit more complicated than that, but that is essentially we're gonna leave the generator alone and only augment the discriminator. And this method is called adaptive discriminator augmentation. And intuitively, what is happening is with limited data, the discriminator gets too good too quickly. So it becomes too good at discriminating whether it's real or fake. It saturates its performance really quickly. And once that happens, you no longer have that information to propagate back to the generator. So it's kind of saying like you're fake with like without any kind of it's kind of like a teacher who says like you're doing good or you're doing bad without any other kind of feedback. And if it consistently saying that you're doing bad, even though the generator is improving, then the generator kind of, in some sense, it gives up and doesn't generate any good information. So you want to kind of balance the performance of the generator and discriminator when training GANs. That's one of the more difficult part of training GANs as well. And doing this augmentation helps with slowing down the discriminator so that the generator has the time to catch up and make better images. And this has been shown to generate pretty good images as low as 1000, which is perfect for our case. Did you have a question? Did somebody, I think I saw it again, but maybe not. Okay. Yes.
You said that you need like roughly 1 million images to get a good GANs.
Yeah.
What proportion of those images can be augmented?
Oh,
That's not a good model.
what proportion can be augmented? So if you have just 1 million generated images, I think you typically people form like horizontal flips or like some kind of rotation or cropping, but nothing like crazy as to change like the color pixel or whatever, because again, you don't want a differently colored images generated. If you have as low, like if you have only 1000 images like this, then that data augmentation is applied to all images, but not at every step. So the adaptive portion, actually, that's a great question. The adaptive portion means that we're going to apply the augmentation, whether it's color or whatever, at a certain percentage of that time. So in the beginning, I forget what it was. So for example, if we set P equals the probability of applying the augmentation itself as 0.1, then 0.1% of the time we will augment it, and then other times we will not. And this probability will actually change while training, hence adaptive. So I think it's actually, we want to do low augmentation in the beginning when the discriminator is still not good. In the beginning, discriminator needs to train, so we don't need high probability to augment, but towards the end of the training, when the discriminator is getting too good, we want to make the task harder for a discriminator, so we will augment more. So we'll increase the P up till P equals 0.9, something like that. Yeah, and the augmentation is kind of done automatically, I guess. They have figured out a way to do this. Yeah. Yes.
Like, how is it like how would this perform, in contrast? Because that 10 to the third images with at the end of the day, 90% being augmented is crazy. Like how what if you just had an annealing term in front of the discriminator or value of the loss, and you just kind of made it not so hard to begin or something? Would that sort of do the same thing? Or?
Yeah, so there's so I guess with the adversarial loss, there's the mathematically, so what it's doing is it's minimizing the KL or Jensen-Shannon divergence between the and one of the properties of the Jensen-Shannon divergence is that unless it's matching, it's all harsh. Like you said, and putting a nailing term works, but there's also other kinds of losses. Like in the lecture, I'll talk about Wasserstein distance, and that one, instead of being all or none, if it's a little bit off, then the loss will actually be lower than being if it's far off, then the loss will be higher. So it will actually scale by how good it will actually give a grade instead of grade of A, B, C, D, F. Why is E good? A, B, C, D, F instead of 100% or 0% kind of. Yeah. So yeah, there are ways to. I think even then, I think the Washington again is actually one of the earlier ones back in 2016 or so. So these are, I guess, even more techniques on top of that. Yes.
For the augmentations, oftentimes you want to pick invariants of your data.
Yeah.
Did you look at different projections of your 3D as a augmentation approach?
That would have been better. Yeah, I mean, I didn't necessarily. Let's see. So one of the reasons why I stuck with this projection was that you could see more information about the neuron. Depending on which an XY, XZ, or YZ you look at, if you look at YZ, it just looks like a clump of strings in some sense and not at least in my eyes, which is not a neuroscientist's eyes, not super informative, and the XY. Is the informative axis in the sense that people, I know in neuroscience, I think there's a way that you can kind of use a 2D view to try to classify cell types, but something like they have like a density plot. They make a density plot of the axon branches, and that's one of the ways to classify, like calculate some of the morphological properties. So that's one of the reasons why I chose that. But yes, I could have technically also increased the size of the trained data set by 3. But by considering all 3 projections, X, Y, Y, Z, XZ, I think the reason why I didn't use that was I still wanted the generator to produce only the XYs only. And if I increase, I guess that in the end, like the generator needs to, I want the generator to produce one image, and I didn't know how to control for how it can generate, how it will choose one of the 3 projections, I guess. That makes sense. Yeah, but yeah. All right, I will move on for now. So I guess we'll definitely talk more. These are all excellent questions that I haven't thought of. Okay, so kind of moving on from the nitty-gritty style yet, I also want to focus on kind of what has been the goal of these generative AI folks in some sense from before diffusion and ChatGPT. It's like, it's not just about trying to generate realistic looking images, but how we can do creative work in some sense. So the literature on GAN is not just about, oh, we're making the most realistic image ever. There's actually a lot more research on how we can control the generation process so that we can make whatever we want to, instead of just what already exists out there in Google Images and whatnot. So, I'm going to show you like 3 examples. This is one of them. This is trying to mix so-called styles from 2 different images. So let's say I have this real image, like source A here, and also some real images, source B here. This with faces. Let's say if I input the source A and add in the style information from source B early on, then actually the generator will generate something like source A in terms of, let's say, lighting or background, but you can see that the gender has changed and the person has glasses. So now, like, so you can think of cases like, oh, like I have these like 2 images that have components of things that I want, and I want something in between or something novel out of them. Then you can, one way is to like mix 2 different styles. So you can see it can change, let's see, gender, pose. It's like this woman is facing towards whatever this woman is facing, and also like put facial hair. You can see if you add in the style kind of later on in the synthesis network, it doesn't affect the original image as much. So you get all the images here have the same pose, like all the people here are looking face forward. But you can see like it takes some other like inspirations from source B. So this one always kind of makes me a little bit laugh because it's a little bit bizarre to me. But so you can choose like the different sources, and you can kind of decide when you want to mix these information to kind of get different outputs. In this example, it's like a fine grain tuning, right? So you have an image of a woman, maybe you created this in Photoshop and you're happy with this, but you want to change just like her hair color or something. Then maybe you can take another image with the right hair color and kind of apply that here. So I mean, these are all kind of cool examples from computer vision. But again, I want to emphasize like, our latent is a gene expression, and now you can think about, okay, can I take components of one gene expression and another and mix them, and like what kind of neural morphology will that come out like? So I guess trying to like apply to whatever your problem is. And I know this is like very fun to look at, but yeah. Some other examples that people have tried is trying to create intermediate images. So in this particular one, the authors found some images of faces kind of looking on one direction, and faces looking in another direction. And maybe you want this face to look sort of towards the center. Then you can try to like take the latents for these and then find the average. You feel like take the weighted average and generate the corresponding image, and you can get some images like these. Not all of them are perfect, but this one has like a part of the face missing, but I would also preface that this was back in 2015 and we can do much better than this. Another a very fun example, and I will have examples for this in the results section as well. I'm not just showing this just because it's cool. Another example is, for example, you have take some images, man with glasses, there's a certain property, so that so all the images of man with glasses, you will know the corresponding like latent vectors. You find the latent vectors of man without glasses. You can do the vector subtraction, like you learned in linear algebra, add in woman without glasses. And if you take that vector and generate the corresponding images, you'll get woman with glasses. So all the women with glasses are generated images, but plausible. Right? So yeah, and I will show you examples of how we utilize this techniques in the results. All right. For the sake of time, I will skip this real quick. Classes. I think I'll be giving GANs and diffusion. Yeah, soon. So I think the class is in dental school. You can ask Yushuan, who is a GSI, on where it is. But yeah. Okay, so jumping to MorphNet. I think I've shown this slide before. So MorphNet uses VAs and GANs to learn the relationship. Once trained, we drop the decoder and we drop the discriminator and just use the encoder to get the meaningful latent representation of the gene expression, and then we feed that into the generator to get morphological information. Obligatory, this works well. Slide. So here, so we train MorphNet on the patch-shaped data set, as I mentioned before. They seem to retain cell type. One, they generate pretty realistic looking neuron images, and they also retain cell type specific morphological properties. By that, I mean, if you take the generated images and try to classify them by cell type, then the classifier can pretty reliably classify them to the right cell type. So that wouldn't be possible if the morphology didn't retain any of the cell type kind of information. When we tested it on the validation set, with the adaptive discriminator augmentation technique, thankfully we didn't have any not any, but we were able to simply reduce overfitting. So the generated images on the validation set still look pretty realistic, but not memorized. So you can see like in the LAM5 case, this neuron looks quite different from what is the ground truth. But you can look at this and say like, oh, but this does still look like a LAM5 neuron. Like this does still look like a VIP, even if it's not exact. So with that, we have applied MorphNet to try to predict morphologies for other parts of the brain region. So with the single cell data sets, we can actually I can combine patch-seq and the single cell RNA-seq from patch-seq and other single cell data sets, which doesn't have morphological information, and then predict from those unimodal data sets. So again, they don't have ground truth. So the middle column here are showing the predicted images. They don't have ground truth because obviously this was just single cell RNA-seq data. On the right, I just provided examples of what these cell types look like. And again, although it's not possible to know oh, this cell, I'm saying that we think that this cell might look like this if we were, but obviously we don't have nobody has done an experiment to go and see. What we can kind of say is okay, it is still generating something that looks like other cells of this cell type. So that's what this two different columns are showing here. Here's one image of the kind of application or the use cases of this is kind of an example of the faces turning from left to right. I did the same thing, except I took a LAM5 cell and then kind of step through some of the intermediate gene expressions to get all the way to VIP type cell. This might be easier to understand. So in the beginning, this is the classification probabilities. So in the beginning, in step one, the classifier really thought that this was LAM5. There's a high probability there. And obviously at the end, the classifier thought that this was VIP. As you progress through, the LAM5-ness goes down and the VIP-ness, if goes up. There's an interesting thing where like in the middle, the classifier is like not sure if this is p-valve or VIP. This is natural, like this is not something that you would see in a biological experiment unless you're trying to like really capture the transitioning cells. So these are just showing like plausible morphologies, that may exist in very like transitory states. Same kind of application. Here we have taken differentially expressed genes from LAM5, remove all of them, so set all the gene count to 0, and added more different values for the differentially expressed genes for VIP. And if we take that and generate the corresponding neuron, the end, so this is the real example of LAM5. If we just manipulate the genes and ask it to produce a VIP, then it looks something, again, not perfect, but it does look like a bipolar neuron, which is kind of characteristic of VIP neurons. Again, this is not perfect because there's probably no cell that has like 0 expression of all the LAM5 genes. That's probably not super real in some sense, but you can kind of see the kind of the manipulation that we can do in gene expression to and see its effect on morphology using MorphNet. Okay, we're almost at the end here. So that was kind of the 2D project. Yes, we are working on 3D. It is a we are actually using diffusion networks for this, and hence I might try to give another tools and tech maybe in a year or so. But here's some like preliminary results. I mean, I know it looks quite similar to the 2D, but these are actually all 3D points that just for visualization purposes, I'm just only showing a 2D portion. Yes.
There's a question in chat.
Yeah, thank you.
Is there a possibility to generate the tree graphs of the morphologies instead of 2G images with some deep learning technique?
Yes, I'm working on it right now. Yeah, you should, if you're interested, please talk to me. I guess. Who is this? Is it possible to know? I'm sorry to put you on the spot.
Right.
Last name RAI.
Okay.
Right.
All right. Please select me. My Yumish ID is Hojae Lee. All right. So I'm going to leave that for a future talk here, and then yeah. So the key takeaways: morphology is a key determinant of neural circuits and cell type identity. We developed MorphNet to predict cell morphology from gene expression. They seem to be doing pretty well, if I may say so. In terms of FID, let's put it that way. FID and just qualitative inspection. We also played around with trying to manipulate gene expression to generate novel morphologies or kind of see what the effect of manipulating gene expression would be. And yeah, Mr. or Mrs. Rai or Dr. Rai, I am working on extending MorphNet to generate tree graphs or 3D morphologies, not just images. All right. Yeah. Happy to take any questions or talk afterwards. I think, sorry, yeah.
Oh, yeah, so do you have like GitHub available for this, or what is the tool?
Yes, I will send that link to Marcy after updating it. I already know that there's like some issues I need to get to on GitHub. It's just I haven't gotten to them, so I want to get to the issues first before sharing. Yeah.
I'm not super familiar with StyleGAN, I guess, but like, how important is it to do, if you go back to the MorphNet model, computation graph. Yeah, like right there, right there. How, like if you were just to encode some gene expression and then you were just to like sample from the mu sigma that you get there and just feed that directly into the generator as the noise, like how important is it to have that noise source there in addition to, you know, the sample mu and sigma from the encoder?
Okay, actually, turns out pretty important because actually the middle portion here is generated with from just trying to transform the noise into like an image. I think there's a little bit of a, if you think about the gene expression vector, we're only taking a 10-dimensional vector and we're trying to create this humongous image of 512 by 512 by 3. I don't think there's like enough information there to just transform directly from gene expression to image. And another key reason is that gene expression is not the entire determinant of morphology. There's other influences. So having the noise actually gives us stochasticity. So actually, I could have generated multiple variations of morphologies from a single gene expression. I'm just showing one example from one gene expression in the paper, and that's a good point. Like it gives the impression that like, okay, this is a morphology for that gene expression, but that's not really the case in biology, right? Like there could be some flexibility. The gene expression only puts a constraint, and the noise actually gives that flexibility to generate different variations. Yeah, and that would be actually a really interesting thing to study, like how much kind of within cell variation how much can it exist within a given like gene expression? Yeah.
Yeah, I mean, well, yeah, I mean, like you could MLP the sample from the AA latent to like
Yeah.
whatever you wanted, but
Yeah.
I mean. Also, I was just wondering, like, so when you do your interpolation, what latent vectors are being interpolated? Are you just doing that? Are you interpolating between some sort of like conditioned GAN noises or between like, how do you get the different cells?
Yeah. So each cell will have the latent representation 10-dimensional, right? So and I'm interpolating in that 10-dimensional space. So I have a 10-dimensional latent vector for a LAM5 cell. I have a latent vector for a VIP, and then literally taking a linear interpolation in that 10-dimensional space.
Okay, and then you're using that to condition the GANs.
Yeah, and then I'm using that to feed the GANs to generate the corresponding morphology. Yeah. Yes.
The images that you're generating are just gorgeous. They're really, really cool. It's fun to look at them all day, but I'm curious if how people typically quantify these images and whether or not the generated images have similar quantification distributions as the real quantified images.
Yeah, that's exactly what my reviewers have said. So that's also partially why we're kind of moving on to the 3D model where the 2D. So I think the only kind of traditional morphometric that you can calculate is maybe shoal profile, which is you start from the soma and draw some circles around it and see how many branches are intersecting, and that kind of gives you the if you draw that profile, it gives you kind of the complexity of the neuron based on distance to the soma. But there's many other morphometrics like branch length or oh my goodness, like a tapering ratio. There's literally almost hundreds of them, and they're not really amenable for calculation from 2D images, which was one of the kind of things that were flagged for. So yeah, which is why we're going to the 3D model as well. Yeah.
So for the shoal,
Yeah.
How close are your generated ones?
I have not computed that, so I will let you know. Yeah. One of the I have not explicitly calculated that. For the most part, I've only kind of quantified how good they were based on the FID metric only, and not the, I guess, in some sense, biological metrics.
And I guess if the cell type is determined from these quantifications,
Yes.
why not just predict those directly?
It's that's an excellent question. So the cell type that was given by Patchseek, I think, was from gene expression, not morphology. So I think they have gotten the single cell RNA clustered and labeled a cluster, and that's where they have gotten the and that's partially the reason why I don't get perfect classification accuracy when I classify the generated neurons. I don't think that's a satisfactory answer, but holistically, I'm thinking that like cell type cannot just be from morphology or transcriptomics alone. It needs to be some kind of combination of the two. I don't know how yet. Does that help? I guess at least, or we can talk more. Yeah, Crystal, yeah.
I don't know whether you have covered this, but maybe I also overlooked this. So you did a very nice work in which that you make use of gene expression information to generate this morphological data. But still, although like gene expression doesn't always equal to morphological information, but I'm sure there are some like shared features. That's how you manage to transfer expression information to morphology. I wonder whether you also try to see whether you can use morphological images that you just generated to reverse generate this gene expression that will closely match the original expression information.
Yeah. I think this is a common question. I haven't done that for for these images, but I know there's even like a recent paper that came out like 3 days ago that there's several papers that deals with, for example, trying to go from histological images to gene expression, something like that.
I haven't came across the papers that when they tend to generate certain machine learning output from one modality. They could also using the same technique to reverse generate to the original input modality. So I wonder whether this could be done with the internet.
Let's see. So I mean, we'll have to probably have another, if I were to do that like right now, I would probably have another network that goes from images to genes. Maybe I'll copy and paste again and then just flip the order. Maybe I'll start from that. What you mentioned, actually, there's a I think she has done this work on like invertible neural networks. Like I think Josh has mentioned this very briefly. CINN is, I don't know what the C stands for. Maybe conditional invertible neural networks, where they had invertible ones and tried to match the latents together after inverting. So that was one of the training methods, so that you have one thing to encode and another thing to be kind of like having two VAEs and trying to have the two latents match each other. Yeah, sorry, I could keep.
Yeah, it's, thank you for your presentation. It's really, really good. You explain a lot of different things. My question is, it was a really good question of yours about the noise.
Yeah.
Used. They said that this introducing the noise will help us account for other reasons, other factors that affect the morphology of the cell. So is there any way to kind of control, like for example, like in style gen, and maybe adaptive instance and normalization, to kind of control this noise and understand other reasons as well, other factors, what affects the morphology?
Yeah. So that's actually a really deep question that we can talk about for like an hour. Yeah, so I have not, I included a slide and then took it out, but I have looked at what MorphNet is learning in some sense, like what are the weights and then how changing that weights affect morphology as well. And that's kind of closely associated with the noise as well. In general, when you change, let's say noise is like a 10-dimensional vector as well, just random. There's a whole slew of research on if we change one dimension, can we change one aspect that we want to control? And this is called disentanglement. But VAEs usually do better on this. So Josh will give a talk, there's a lecture on disentanglement. Basically, for example, for the faces, I'll just make the analogy. Let's say these are all coming from 10-dimensional vector. Can we change the first dimension, let's say from 0 to 50, and then change the age of that person, from young to old? And most of the time, VAEs or GANs, they change multiple aspects. So if you change one dimension, it will change the gender, it will change the age, it will change the hair color, like all at the same time. And so these properties are entangled together, but there are ways to make it so that the properties are separated out. That way we can also kind of control, like, can I just change one aspect of the noise so that I make neurons that are bigger or longer or more branchier or something like that.
Yeah, like it's called that.
Yeah.
How do you pronounce Salgan?
Salgan, yeah.
Salgan.
Yeah.
And the second question is, what are the main struggles and hardships with doing the 3D?
3D, I guess it's just, in some sense, it's a different problem. We're not generating images anymore. So what deep learning architectures can do very well with handling kind of tabular data, that's been the first kind of challenge. Like, what can we use to generate points? I know there's like things like point nets or like using point clouds, but they are not as, in some sense, mainstream as GANs or SAL-GANs. The other thing is we can generate points, but we need to connect them together into a tree. So how do we connect them together? That's another question. How can you put, so we've actually approached it in multiple different ways. In the beginning, I tried to use graph neural networks because you can think of them as, so I can predict both points and the edges that connect the points together. That did not turn out very well. So now I'm using a different approach, which is modification of a diffusion model. Yeah. Yes.
There's two questions online, and so I'm gonna ask those questions, and then we are over the hour, so I think we'll have to wrap up. after.
Yeah, feel free to have more pizza, leave, go to class, I don't know, do whatever you need to do.
Okay, so the first question is from Cedar, who said, I noticed that the F1 scores and ACC are ratio to real scores. What is the real scores on real data set and generated data set? Is it a classifier? It's not.
Yeah, excellent. I don't know if you finished or, yeah, I didn't understand the question. Yeah. So, classifying these kind of images, even on real, pretty hard problem. You can probably write a paper about that one. I try to make the best classifier that I can, which could have been a paper on its own, but it's about like 70% or so. So, when I say like in relation to the real, that means that, if the real classifies this accurately, like 70% of the time, then, the generator will classify like a little bit less than that, maybe 60% of the time. That's why, yeah. Even though it's just like six-way, there were 6 classes in this, even though it's like six-way classification, it is hard to classify.
Thank you, and then I'm gonna try to read the second question. Hopefully I get through it a little better. So Arvind Rao asked, said, nice work. It's cool you can subtract gene expression selectively. If you subtracted the Yamanaka SC factors from the neuron, would you get a pluripotent SC? And then if you added it back, can you regenerate a differential neuron stem cell?
That is, I haven't tried that like in particular, so I can try to do that much now, honestly. I'm kind of curious now. But that's the whole point, kind of the application of this, like, right. We want to try to see like which genes we can knock out and add back in and see how that affects morphology. The only thing I guess I know is that I can't just knock out one gene. There's no like one silver bullet gene that will like destroy the neuron or not. Like usually I have to knock out like several kind of classes of genes kind of to have a significant effect on morphology. Yes.
The other factors don't like, I mean, it's a cascade, right?
Yeah. So I don't know if I don't know if the question was like knocking out one particular or like a class, but if it's a class, then yes, that we'll definitely see some effects. I will have to actually do it to see the effects. If it's like one gene, probably they'll have no effect.
Arvind followed up that there are about 20 Yamanaka stem cell differentiation factors.
Okay.
And then
I think that should be enough. Yeah. Yeah, but that's a simple thing for me to run. It's just I haven't done it. That's probably not a good answer.
So real quickly, the Cedar did also ask how to evaluate the 3D images.
Because with 3D we can calculate a lot of the morphological metrics, like I said, like branch length, like total area, total volume. So we can actually compare the 3D on the biological metrics, and I still need to look into what is a good metric for like something like the FID, but for 3D that sure exists, but again, I think it's still under research what that might be. Yeah.
All right, well, with that, thank you so much. Hope to see everyone next week.